A nerve impulse is generated when the stimulus in an environment is strong or when it is stimulated by another neuron. For example, if someone were to step on a sharp object like a tack, then an impulse would be triggered. The part of the neuron that initially senses the stimulus is called the dendrite. Dendrites are needed for the cell to become active and are responsible for conducting the electrical messages that get sent to the body of the cell. The body of the cell then transfers the electrical message to the axon of the neuron, where chemical and electrical changes occur. Before the neuron receives the electrical message that causes the electrochemical changes, the neuron is at resting potential. This means that the inside of the cell is negative and the outside is positive. The charges in the cell come from sodium and potassium ions. Sodium ions linger outside of the membrane, making the charge positive, while the inside of the membrane holds potassium ions, making the charge negative. The sodium-potassium pump, along with ion channels, allows for sodium and potassium ions to travel across the cell membrane when their respective gates open. Once the electrical message reaches the axon, the voltage-gated sodium ion channels are triggered and open, allowing for all of the positively charged sodium ions to rush into the membrane, causing the cell to become depolarized. The depolarization of the cell causes an increase in voltage, triggering an action potential to occur and keep the electrical message moving across the axon. After the depolarization process goes down the axon, then the repolarization process begins. This time, the voltage-gated potassium ion channels open, allowing the potassium to flow out of the cell membrane and in an attempt to rebalance the charges. The sodium-potassium pumps then take over to bring things back to their resting level, and once all the ions have been restored, then the recovery phase is complete. Once the electrical message travels across all of the axons in the neuron, it will then make its way to the axon terminal, where it will go through a synapse that converts it into a different type of signal so that it can be transferred to the next neuron. The axon terminal is a part of the presynaptic neuron that is sending the signal to the new neuron. The axon terminal holds synaptic vesicle sacs loaded with molecules of neurotransmitters. The receiving cell, called the postsynaptic neuron, accepts the neurotransmitters in its receptor region. Between the two neurons, there is a tiny gap called the synaptic cleft. After the electrical message races through the axons, it comes to the axon terminal and triggers voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. The flow of the positively charged calcium ions causes synaptic vesicles to fuse with the cell membrane and release their neurotransmitters. The transmitters then flow across the synaptic cleft and bind with the receptor sites on the postsynaptic neuron. Once the neurotransmitters finish transmitting the impulse to the new neuron, they travel through uptake pumps that reabsorb them into the presynaptic neuron to be used again for the next synapse. During this process, the first neuron has converted the electrical signal into a chemical one. However, in order for the signal to become an action potential again in the receiving neuron, it has to be converted back to electrical. This happens when a neurotransmitter binds to a receptor, because it's what causes ion channels to open. Depending on what transmitter binds to which receptor, the neuron might get excited or inhibited based on what the neurotransmitter tells it what to do. In an excitatory postsynaptic potential, excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to an action potential, making it more likely to continue the message on to the next neuron. This creates divergent nerve pools. In an inhibitory postsynaptic potential, an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, causing the charge to go down away from an action potential, which makes it hard for the signal to continue. This causes convergent nerve pools. The likelihood of the postsynaptic neuron developing an action potential depends on the sum of all the excitations and inhibitions in that area. So far, the body's response to the stimulus has been handled by the sensory and afferent neurons in the sensory division of the peripheral nervous system, which is responsible for obtaining data and sending it to the central nervous system. But now, the responsibility is passed because the pain signal from the tack has triggered an action potential in a neuron in the spinal cord, and there it reaches a point where the motor and efferent neurons take over in the motor division of the peripheral nervous system. Once the signal is interpreted by interneurons in the brain, the message is transmitted to motor neurons, which send an action potential back down your leg where it reaches an effector that reacts to a motor neuron signal. The motor neurons then allow for a foot-lifting response until the rest of your body is able to figure out what happened. All of these steps combined produce a reflex arc or your body's reaction to the stimulus.